programming and specifically in the context of early exposure to conflict between parents. Next, I'm going to talk about resting state functional connectivity, MRI, as an important means of studying early brain development with links to subsequent mental health. And lastly, I'll present results focused on a specific functional brain network, the default mode network, and looking at it as a potential mediator between exposure to early life stress and behavioral health. So interparental conflict, or conflict between parents, is a common form of environmental stress in homes with infants and young children, but it's not benign. It's repeatedly been associated with mental health problems, including depression and anxiety, and these strong connections between conflict and mental health outcomes exist regardless of socioeconomic status, sex of children, or ethnicity. And there's evidence that vulnerability to the effects of conflict may be shaped early on during infancy, during a time when aspects of the nervous system that are tightly linked to mental health are rapidly developing. And this idea that times of rapid development of aspects of the nervous system, they may be particularly vulnerable to environmental influences and to potential disorganizing effects is known as developmental programming. So we previously looked at levels of interparental conflict in the home during the first year of life and infants' neural reactivity to a stimulus that we think is relevant to conflict, and that is angry tone of voice. And we found that infants in homes with higher levels of conflict showed greater reactivity to very angry tone of voice in brain regions that we think might be important, especially that we know are important later on anyways, for stress and emotion regulation, and particularly the rostral anterior cingulate cortex. And so these findings suggest that potentially early exposure to conflict may be associated with the functioning of brain regions that are important for mental health later on, and potentially that could influence the vulnerability in the subsequent environment. And I think more generally, these findings also speak to the potential for fMRI with infants to be sensitive enough to detect variation in brain activity that's associated even with a moderate form of environmental stress, like non-physical conflict between parents. But these findings only speak to brain reactivity in response to a specific stimulus, which is angry tone of voice in this case. They don't speak to brain functioning more generally or to how brain regions might be functioning in a coordinated manner. So for the brain to function in a healthy manner, it needs to be responsive to input. And task-based fMRIs in the previous study tells us about this responsiveness to input. But healthy brain functioning also requires communication among brain regions in order to integrate information. And in order to do this, the brain forms functional networks. And resting state functional connectivity MRI can tell us about these functional networks. Thinking about development of these functional networks and potential influences of early life stress during infancy is particularly important because there's been some recent literature that suggests that these functional brain networks begin developing very rapidly during the first year of life. Several studies have shown this now. And this rapid development during the first year of life, I think, speaks to potential for developmental programming during this period. So one of these networks that has been one of the most well-studied that develops rapidly during the first year of life is the default mode network. It's shown to develop rapidly, particularly during the first year of life, and this is even in studies that are looking from 0 to 2, showing that the first year may be really particularly important for the organization of this network. There's also evidence that this network is impacted by severe forms of early life stress, including preterm birth and childhood abuse. And another reason that we're interested in this network is that it's been implicated in mental health outcomes later on, in multiple mental health outcomes, but for example, depression and ADHD. So despite this compelling work about the development of the default network and its implications for mental health, we really don't know any about associations between the development of this network and common environmental inputs during the first year of life. And we also don't know, despite the associations with mental health later on, we don't know about associations between this network and behavioral health during infancy, whether there are some of those connections or whether some of those associations might be emerging earlier on. So this study had three main questions. The first was whether the level of interparental conflict in the home during the first year of life would be associated with infants' default mode network connectivity. The second was whether the strength of connectivity might be relevant for infants' behavioral health, and specifically for negative emotionality. And then finally, we wanted to see whether the strength of default mode network connectivity might mediate between the level of conflict in the home and infants' negative emotionality. So this was a sample of infants living in their homes with their biological parents, and we screened for level of conflict to make sure we would get a range of conflict in the sample. We also excluded for any involvement of Child Protective Services because we didn't want to have infants who were exposed to more severe forms of stress or physical aggression. We assessed conflict with the revised conflict tactic scale. Anything obnoxious on this slide is the result of my advisor. And so we assessed conflict with the revised conflict tactic scale. We had mothers fill that out. We assessed infant behavior with the infant behavior questionnaire and specifically focused on two subscales that form a negative emotionality composite. Okay, so scans were conducted during natural sleep at infant's bedtime. The study focuses on a 6-minute resting state scan. And I will note that after removal of frames for motion, all scans were still greater than 5 minutes. Okay, so to answer our first question about the association with conflict and DMN, or default mode network strength, we used the posterior cingulate seed region, and if anyone has questions, I can talk about why we chose that. And we did a whole brain regression of conflict on the posterior cingulate seed region. Next, to look at connections with infant emotionality, we extracted values for the strength of connections that we identified in the whole brain regression, and we then looked at associations with negative emotionality. And finally, we ran a model to test for potential mediation for connections that were both associated with conflict as well as with infants' negative emotionality. Okay, so these are the results of the whole brain regression with the posterior cingulate seed region. And you can see that higher levels of conflict were associated with greater connectivity of the posterior cingulate to the rostral anterior cingulate cortex, or anterior medial prefrontal cortex, whatever you want to call it, as well as additional default network regions, including the inferior temporal gyrus. These are the subcortical results of the same analysis, showing that higher levels of conflict were associated with greater connectivity between the posterior cingulate and the right amygdala. So now moving on to extract correlations and look at associations with infants' negative emotionality. So we focused on two connections. We focused on the connection between the posterior cingulate and the anterior medial prefrontal cortex, as well as the posterior cingulate to the right amygdala. We did that because there's previous literature suggesting that these connections may be vulnerable to stress, as well as important for mental health outcomes. So the posterior cingulate to the anterior medial prefrontal cortex we saw was significantly and positively associated with negative emotionality, so stronger connectivity between those two regions is associated with higher levels of negative emotionality. Similarly, for the posterior cingulate to the right amygdala, we saw a positive association with negative emotionality in infants. And you can see from this slide that the posterior cingulate to the anterior medial prefrontal cortex, that association with negative emotionality was stronger. All right, so next to test these connections as potential mediators between conflict and negative emotionality, we entered both of those connections in a mediation model. And what we saw from that was that only the posterior cingulate to the anterior medial prefrontal cortex connectivity mediated between higher levels of conflict and infant negative emotionality. So another way to say this is just that higher levels of conflict, there seems to be an indirect path from higher levels of conflict going through this connection to infants' negative emotionality. So broadly speaking, I think that these results speak to the potential sensitivity of resting state functional connectivity MRI to be associated with moderate variation in stress in the environment. And in terms of the specific connections that we identified, they are consistent with previous literature, thinking about what connections might be vulnerable to stress. But I think they extend this literature by moving it down to infancy, as well as looking at a more moderate source of stress. Thinking about the posterior cingulate to the amygdala, the amygdala is of interest because in the world of early life stress literature, this region is really a frequent focus, thinking about potential mediation for effects of early life stress on subsequent emotional problems and mental health. But these findings kind of extend that to human infants. That's been shown with animal literature as well as with older children, and these findings extend that down to infancy and provide some initial empirical support for an association between early life stress, connectivity of the amygdala, and infants' emotional functioning. Thinking about this posterior cingulate to anterior medial prefrontal cortex connection, one thing that we think is important about this is both of these regions have been shown to be hubs or highly interconnected regions of the default network. So it's possible that anything that might influence the connectivity of these two regions could have implications for development of the broader network over time. In addition, anterior to posterior default mode network connectivity has been in adults repeatedly associated with depression, and specifically with a repeated focus on negative emotions. I don't think that these results speak at all to risk for depression, but they do show a potential association with the anterior to posterior default mode network connectivity and emotionality beginning in infancy. So one thing I wanted to mention was about the direction of the effects, which at first were a bit surprising to us. Previous work, this is on the right here, is an image from a paper by Smizer and colleagues, showing that this is showing that term infants show anterior to posterior default network connectivity, but they don't see this connectivity in preterm infants who are scanned at term age equivalent. And so the implications of this paper are that preterm birth, this stressor, would disrupt this anterior to posterior default network connectivity. In contrast, we showed that higher levels of conflict in the postnatal environment are associated with greater connectivity between these two regions. And we also have some, and I think what I want to say first is that I think maybe the discrepancy in these findings has to do with timing. The stressor they're looking at really can be considered to be during the prenatal developmental period or prior to gestational age equivalent, whereas our findings are postnatal. And we have a poster later today that looks at the vulnerability of this connection to another form of prenatal stress that also suggests both its vulnerability and potential effects of timing. And this is just a few other little things I was going to say about the importance of timing and how rapidly prenatal and early postnatal brain development is occurring. And because of this rapid development, I think that it makes sense that different forms of environmental input or stress might have very unique impacts depending on whether specific periods during the prenatal as well as the postnatal environment. And this graphic here is actually showing a pretty dramatic switch in gene expression in the brain from the pre to postnatal environment, suggesting maybe some really dramatic changes in the neural landscape from this pre to postnatal period. I think that could result in very different effects of environmental inputs. Okay, so lastly, I think these results, again, just speak to the potential for using this methodology to understand brain development in the context of various forms of environmental inputs. There are lots of future directions for this work. I'll just mention a few, one of which is that this is a snapshot and longitudinal work and looking at network development over time, as well as stressors over time is going to be a really important component of future work. We also recognize the default mode network is one of many functional brain networks, and we'll be looking at additional networks as well as global functional topology. More in-depth measures of environmental stress as well as behavioral outcomes are also going to be a really important component of future work, as these were both based on questionnaire measures. are environmental stress and behavioral outcomes in this study. And lastly, a really interesting and exciting thing about resting state is that it can be conducted in animal models also. And I think the use of those models are going to help us move towards a better understanding of potentially causal effects of these environmental inputs on network development, as opposed to sort of the correlational studies that we're able to do right now in humans. So I wanted to thank the FAIR Lab, and particularly Damien and Mark Rudolph, as well as my grad school advisors, Phil Fisher and Jen Piper. Thank you.
Jared, come on up. And while he's getting prepared, Alice, we can take one question. Yeah.
Fascinating, great talk, fascinating research. I have a question and a comment.
Yeah.
My question is, do you have any sense or assessment of whether there's actually this maternal stress going on in families that have higher conflict? Like, presumably, you would think if they're having conflict, after birth, that it might have been going on during pregnancy?
Oh, yes. I definitely think it's going on during pregnancy. And I didn't get into this at all, but we retrospectively asked about pregnancy conflict and controlled for that in these analyses. And we have some suggestion. I don't think we can really get into it because it's a retrospective assessment, but that we would see that opposing effect, that pregnancy conflict might have an opposing effect on the default network versus conflict postnatally.
Okay, interesting.
That makes sense. Yeah.
And then my other thing for the directionality, we know that there's very different stress neurocircuitries for physical stressors versus psychological, and then predictable versus unpredictable. And in the NICU, they usually pair everything together, and it's kind of at the same time of day.
Yeah.
So it's kind of a thing to explain the opposite direction.
Thank you. Yeah, and I think work in the NICU and with preterm infants is a really important direction for this work.
Thank you.
Yeah. Thank you.
All right. Okay, so next up we're going to have Jared Rasmussen from UC Irvine.